which is actually a change from the printed calendar. Our first speaker will be Robert Verity from the Imperial College London, speaking about variation at the var2csa locus, results of a cross-sectional study from the Democratic Republic of Congo.
There's your pointer. Forward is the big green button, and the laser pointer is the green laser.
Okay, thank you very much. Thank you to the organisers for allowing me to speak about this today. And before I start, I just want to acknowledge that the work I'm presenting today is just a very small part of a much wider project that's been ongoing across several institutions and over several years. So I can't take credit for too much of this work. And I'm very grateful to everything that's been done at these institutions, in particular thanks to the Meshnik and Giuliano labs at UNC Chapel Hill. So one of the overall aims of this wider program of research is to try and understand malaria epidemiology and population genetics in the Democratic Republic of the Congo. So the DRC is a very important country in terms of malaria epidemiology. It's a very large and diverse country. It's right in the center of the malaria belt in sub-Saharan Africa, and it has one of the highest malaria burdens of any country in the world. And yet, historically, there's actually been very poor data, both epidemiologically and genetically, from the DRC. The two really high-quality surveys that we have are two demographic and health surveys, or DHS surveys, one in 2007 and then one in 2013 and 14. And these are national, cross-sectional, large-scale surveys. In 2007, there were 300 spatial clusters, with a cluster roughly corresponding to the village level. And then in 2013 and 14, this was almost doubled in size to 540 clusters. And there are a number of other advantages to the DHS data. Because it's cross-sectional, we get a kind of unbiased slice of the parasite population at that point in time. We get to see asymptomatic as well as symptomatic individuals. It's also a very well-designed and balanced study, meaning it's nationally representative. We have a very high spatial resolution as well. The DHS questionnaire includes a malaria section with 42 malaria-specific questions. So we have a large number of covariates for any one of these clusters. And children under 5 years of age get heel or finger prick blood taken, which is used to prepare slides for microscopy, plus an RDT, plus dry blood spots. So these two DHS studies have really filled in a lot of the gaps in terms of malaria epidemiology in the DRC. And these dry blood spots also may be the key to understanding parasite population genetics in DRC. So these can be sent away for DNA extraction and sequencing, giving us kind of a slice of what's going on in the parasite population at those two critical points in time. So I'm going to talk about two projects that have used the DHS data in this way. One is the variation at the var2csa locus as advertised, and I'm also going to mention some very preliminary results we have on DHPS mutations, which show a very contrasting pattern to the var2csa. So var2csa is one of around 60 VAR genes that are involved in antigenic regulation in the parasite. var2csa is particularly important in pregnancy-associated malaria. var2csa encodes the var2csa surface antigen, which is expressed on the surface of infected erythrocytes, and it allows them to bind chondroitin sulfate A, which is found in the placental microvasculature. This in turn causes infected erythrocytes to sequester in the placenta, and this leads to some of the adverse effects of pregnancy-associated malaria, which include an increased risk of stillbirth, preterm birth, low birth weight, and also increased risk of severe anemia to the mother. Infection also triggers an immune response, and this means that women are at greatest risk in their first pregnancy and risk declines in subsequent pregnancies due to the building of an immune profile. And for this reason, there's been a lot of work looking at var2csa in terms of designing a vaccine against pregnancy-associated malaria. And this is really focused in on one particular region of the protein, which is the ID1-DBL2X domain. And this is known to be the minimal region that can elicit an immune response that's as strong as the full-length protein. At the actual DNA sequence level, there's again been a lot of really good work. One study in 2014 looked at global variation at var2csa, and it found 260 variants among just 247 samples. So there's really unprecedented variation at this locus. They found that 1/3 of sites were polymorphic, and 83% of changes at the DNA level also impacted on the protein sequence. So this is functionally relevant variation. And interestingly, they also found extensive sharing of variants over very large geographic scales. So although there's very high diversity, it appears to be similar diversity throughout the globe with very little differentiation. So more recent work on the kind of phylogenetics of the gene has identified anywhere between 2 and 5 clades to the sequence. And these can be characterized roughly as a 3D7-like or FCR3-like clades. That's one of the main divisions. And these have recently been found to associate with a different risk of low birth weight and being small for gestational age. So in summary, var2csa, it's very important in terms of vaccine design against pregnancy-associated malaria, but it's also just very important in understanding the fundamental epidemiology of pregnancy-associated malaria. So we set out to try and characterize variation in the DRC using data from the 2013 DHS. Dry blood spots were sent away to UNC, where DNA was extracted and tested for parasites by qPCR of PFLDH. We then subset down to 115 clusters of the total 540, and these clusters had to each contain at least 3 parasitemic children, whilst trying to maintain that spatially representative sampling as much as possible. DNA was then pooled in equimolar volumes in each cluster to ensure that different parasite densities in children all contributed the same amount of DNA. And this was then sequenced for a 400 base pair region within the ID1 DBL2X domain, which was amplified and sequenced on Illumina MySeq. And finally, haplotype determination was carried out in the program SeqDeep, developed by Nick Hathaway. I should point out, I'm going to put all my kind of, I'm going to put all my references and links at the end so that people can have access to those. So this allowed us to ask 3 key questions. First, what is the level of sequence variation in DRC? Secondly, is there any spatial structure to that variation? We have very fine spatial structuring in these clusters, so we could really ask that in quite high detail. And thirdly, are there any epidemiological covariates that can help explain the variation that we see at var2csa? So first of all, in terms of sequence variation, we identified 583 sequences in the 812 kids that went into those clusters. So very high sequence variation, although that's actually proportionally lower than what's been found in previous studies, which kind of maybe suggests that we're starting to reach saturation of that rarefaction curve. We also blasted every sequence individually against NCBI, and what I've shown here is the blast percentage of the top blast hits against the number of clusters that the sequence was found in. And you can see that our most common variant was actually 100% matched to a previously identified sequence from Senegal. And in general, there was a strong correlation that variants that were found in a large number of clusters also tended to be similar to sequences that had been seen previously, which is suggestive that common variants in DRC are similar to common variants in other areas. We then attempted multiple sequence alignment. This was made extremely difficult by 2 hypervariable regions that have been, these have been described before. These contain a large number of insertions and deletions, which made it very hard to align. When we eventually did get an alignment we were happy with, we decided to kind of discard all the information in these highly gappy regions, which is throwing away some information, but it gave us more confidence in what we were left in, that we could call polymorphisms correctly. In these remaining sites, we found that 1/4 of sites were polymorphic, so very high variation, but again, slightly lower than previous studies, and a Tajima's D of 1.95, indicating, suggestive of balancing selection. When we constructed a phylogenetic tree using simple neighbor joining, we found very little structure whatsoever, just this explosion of variation. I've not shown any bootstrap values, but they're also extremely low on this tree, so there's very little phylogenetic structure in this particular part of the gene. Moving on to the spatial results, we found no signal of simple isolation by distance. This regression of genetic and geographic distance was non-significant. And we also carried out a more subtle analysis looking for barriers and corridors to gene flow. So there was a talk on Monday about the program EAMS, which was a very nice way of trying to identify these barriers and corridors. I'd just like to bring MAPI to people's attention. I believe this even has one shared author to the EAMS paper. This tries to do something very similar, although it's not as sophisticated, but it's just doing a visualization and permutation testing to identify significant areas of high or low connectivity. So high connectivity here would be blue areas and low connectivity would be red. You can see that we found this very little, very few barriers or corridors to gene flow, so again, very little spatial structure to this variation. The modeling in terms of epidemiological covariates, we constructed a series of general linear models trying to predict allelic richness, so the number of variants per cluster, as a function of prevalence, pool size, CT value, which roughly corresponds to the amount of DNA, and then the DHS covariates of province and urbanicity, along with all second-order interaction terms. And we used backwards stepwise model selection to try and identify a minimal best-fitting model. And our best-fitting model was actually very simple. It included just 3 out of those 30 predictors, including a first and second-order term, a prevalence term, and then a small but significant effective sample size. So in general, the best fitting model had predicted a very strong relationship between prevalence and the number of var2csa variants in a cluster. You can see the true data here are the black points, and then the pink kind of ribbons are the model fit. So it's capturing that trend, and it's also capturing the variation fairly nicely. So just to summarize the var2csa findings, we found evidence that var2csa is under balancing selection in DRC, which again is exactly what we'd predict from a gene involved in antigenic regulation. It showed extremely high variability, although the same common variants were found in many clusters, which again matches what's been found previously at this locus. There was very little phylogenetic signal. There was very little spatial structure, although we did find that variation correlates with prevalence. So we were kind of fairly happy with these findings. They made perfect sense in retrospect, as things often do. We were slightly disappointed with this lack of spatial structure, because one of the advantages of the DHS data that we've got is we have really high spatial resolution, and yet at this particular locus there just wasn't any signal. So that was a bit of a waste of that data. So I just want to contrast this against some very recent work that's just come off about DHPS mutations. So these point mutations at DHPS confer resistance to sulfadoxine. Point mutations at DHFR confer resistance to pyrimethamine. And these two things together are implicated in SP resistance, which is very important because although this is not the frontline treatment in DRC, it is still given as part of intermittent preventative treatment in pregnancy. And we also believe it may be given in fairly high quantities in the private sector. DHFR mutations are known to be at high frequency in DRC, and therefore DHPS mutations are very suggestive of SP resistance. So again, we used the same DHS data, this time 608 samples in 301 spatial clusters, and these were sequenced from Jeff Bailey's lab using molecular inversion probes, which is something I'll come back to in a minute. So these probes targeted 28 neutral microsatellites, a suite of drug resistance loci and antigenic loci. We haven't finished analyzing this yet, so I'm just going to talk about the DHPS mutations. And again, haplotype determination was carried out in seq-deep. So we are going to focus on the K540E and A581G mutations, which are 2 point mutations in DHPS known to confer resistance. And we just asked the question, is there significant spatial clustering? And if so, then where are those mutations? And this second question we tried to answer using the program PrevMap. So again, this is a very nice package that I'd like to draw people's attention to. This is an R package from Peter Diggle's group that fits Gaussian process models. So it's kind of like one step up from traditional kriging because it allows you to fiddle your parameters by maximum likelihood or Bayesian methods. It's a very, very nice package. And this is what we found. So the K540E mutations strongly cluster in the northeast of the country. You can see that, I don't know if you can see the stars versus the open circles here. The stars are clusters where at least one mutant was found. So there's really very strong spatial clustering. The A581G, intriguingly, have the same distribution but occupy a smaller range. And in fact, whenever we found A581G, it was always against the background of K540E. And both of these were very significant in permutation testing for spatial clustering. So just to summarize quickly before I go over time, just going back to this general and fairly vague aim of understanding epidemiology and population genetics in DRC, just to make the point that there is no one story when it comes to parasite genetics in any given area. Each locus tells a different story, and therefore what we probably need is to sequence maybe a large portion of the genome, or at least think about different parts of the genome separately. Fortunately, it seems like it's becoming ever easier to get large numbers of loci. Just to track a trend in increasing data in the Meshnik lab from 2015 to 2018. In 2015, we had 7 clusters and 5 microsats. I've shown something in the middle. And then in the near future, we're hoping to get samples from 900 adults in 2007, 900 in 2013, at 1,800 SNPs. And this huge increase in the number of SNPs is made possible through these molecular inversion probes. So just finally to draw your attention to a symposium tomorrow morning at 8 o'clock, which is going to go through these in detail, and I've heard it's going to be worth waking up for. Okay, so thank you to all my collaborators. Thank you to the Malaria Gen and PF3K project for all the groundwork that we're building on here. And I'll take any questions. Thank you.
Any questions? There's also some seats down front if people in the back would like to take a seat.
If there's no questions, I'll ask a question.
Okay.
So you have the var2csa SNPs. Have you looked at neutral SNPs to see whether they structure differently?
So we've looked at kind of DNDS ratios within the gene. We haven't looked at kind of neutral SNPs surrounding the gene. We do have a series of microsats, but we don't think any of them are close enough to actually be significantly linked. With such high transmission intensity, you really have to have loci that are very, very close to the gene in question to see something like that. So no, but we're hoping to look into that in the 1800 MIP data set. Yeah.
Okay. All right.
Thank you. Oh.
You indicated something about the consistency and diversity that's seen in var2csa across the globe. So in this data set that you've looked at, you have these nice alignments that you're confident of. Have you been able to start to look at sites of convergent evolution where you might be able to map those onto immune correlates of what's being recognized?
Yeah. So we haven't directly done that kind of stuff. I think there is a lot of very good work that's ongoing in that area, and it's very important work. One thing that I've kind of come to the conclusion to is that traditional phylogenetic methods might not be the best way to go actually with var2csa. That the approach that we took of trimming down to confident areas and then doing traditional phylogenetics, we ended up losing a lot of information. It may be that completely different approaches may be better in terms of joining networks and trying to use this pattern of indels as well to try and draw information. Yeah, so that's kind of where we're hoping to go in future with it.
Thank you very much.